My name is Martin. I'm an application scientist at Araceli Biosciences. But the main talk today is been given by Fernanda, who is a postdoc scientist, biologist by training, and doing a lot of data science right now at the COMAS Screening Institute at the Max Planck in Dortmund. So please, welcome, Fernanda.
Hello, everyone. Thanks a lot for being here today. I also would like to thank Araceli and Martin for the invitation. And so today I'm going to talk to you about the cell painting on the Araceli Endeavor system and some insights about that. So as Martin said, we work at the Compound Management and Screening Center at the Max Planck Institute in Dortmund. And this screening facility is led by Dr. Sonja Sievers. And I would like to say just a few words about that. So there we do two types of screening. The first one is a target-based screening, where you usually do some biochemical assays. You perform some hit selection, and then you look at the cellular effects that are caused on the cells. And the other method is the reverse chemical screening, where you have a phenotypic screening, such as cell painting. And then after you find your hit, you identify your molecular target. So those are two valid approaches that we are using at COMAS. And there we also have a big compound management center, which is led by Karina Zeitz. And we do have more than 60 collaborators all over the world. They can all submit their compounds to COMAS. And we do have 25K compounds from these collaborators. We also have more than 100K HTS diversity library, as well as commercial libraries, chemical probes, natural products, so all sorts of compounds, which we can test using these platforms, these two robotic platforms for high-throughput screening that we have. And we just, in February, acquired the Endeavor system, and we are now preparing to implement it into our robotic system this summer. But we are already using it at the bench a lot. So I'm going to talk to you about the cell painting, how we do the cell painting at COMAS, then how we perform this benchmark with the Endeavor, the new system, and finally the outcomes. So the cell painting assay at COMAS is performed with the classical cell painting dyes. So we are using the 6 dyes, which stains 8 organelles inside the cells. And then after we acquire the images with our system, we do the segmentation, the feature extraction, and of course the profile, the morphological profile. And with this morphological profile, we can generate these profiles for a lot of different reference compounds, which we can use to compare to new compounds, which has unanticipated mechanisms of action, right? And then we have also clustering methods where we can put our new compounds and find what they are doing on the cells. And the analysis pipeline was developed by our former member and father of cell painting workflow, which is here today. So we thank Dr. Axel Paul a lot for that. And yeah, so on this talk, I'm going to talk about, I have 3 questions, And so the first one, if CellProfiler version affects our analysis. And this is because on our current workflow, we are using CellProfiler version 3. And we want to move to the newest version, also to update our pipeline. And of course, because the Endeavor images, they are quite big, so it goes well with the newest version of CellProfiler. Also, we want to understand if the clustering with the 4 channels are enough, since our old system has 5 channels acquisition and the Endeavor has 4. And also if the feature set should stay the same, because we do have a fixed feature set and we want to see if this is working also for Endeavor. So I'm going to talk about the details of the cell painting now. So we are doing these assays with U2OS cells. They are epithelial cell derived from osteosarcoma, and they are extensively used in research. And we plate those cells using an automated equipment, and then after 4 hours of incubation, we treat those cells. And to do that, we are using the Echo liquid dispenser, which can dispense really small amounts of the compounds. You already know that. And which is nice that we can do is treat one 384 well plate with 352 compounds per plate and 32 DMSO controls. And we can generate 3 replicates with these plate layout shifts. And these shifts, as you can see here, the DMSO control here in yellow, and then is shifting along the replicates. And this is quite nice regarding batch effects. So with that, we can avoid a lot of batch effects. So after we added the compounds, they are going to stay on the cells for 20 hours. And then we can proceed with the cell painting staining. So we are staining the nuclei, the endoplasmic reticulum, the actin, Golgi, plasma membrane, mitochondria, and RNA. So a lot of organelles. And then we were performing the acquisition with our prior imaging system. And it would take 3.5 hours per plate. So we usually do 12 plates every time we run a new cell painting dataset. And yeah, this is quite time consuming. And now that we have Endeavor, this goes down to 7 minutes. It's insane. So really cool that we can get this time down now. And which is also nice is that the prior imaging system, we were acquiring 9 sites for each well, and this covers 4.6 square millimeters. And now with the new system, we only with 4 sites, we cover almost 6 square millimeters. So this is quite good. We are now having more cells also on our pictures. And as you can see here, this is the Endeavor field of view, and here our prior imaging system. And yeah, it's quite different. We are really acquiring more cells with the Endeavor and with the same or better resolution. And if you do a zoom also on the grayscale images, here I was showing the overlaying of all the channels. And here, these are the channels in grayscale, and you can see the DNA, the ER, the endoplasmic reticulum, here the actin, Golgi, and plasma membrane, and here the mitochondria. And one thing that is different in our prior imaging system is that we were acquiring RNA in a separated channel, in its individual channel. And this is not happening anymore with Endeavor, but then we are going to discuss that on the results, how this affects. Okay, so after we did the image acquisition, we can go to analysis. And this usually takes 6 hours per plate. We do run on an internal cluster. And the way that I set up the analysis is by doing one plate after the other. And then I start the CellProfiler for each CPU that I have available to analyze each image. And yeah, this is actually the bottleneck now, because we do this in 7 minutes, but then the analysis takes 6 hours. So yeah, this is what I'm also working on to enhance and make this faster. Yeah, so CellProfiler, we can configure a lot of modules. And what we're doing here is segmenting the nuclei, the cell, the cytoplasm, and then extracting all the features that we can for every channel and every object on the cells. And finally, those features, they can be combined into a database. So talking a little bit about the set of features that we have, comparing CellProfiler 3 and CellProfiler 4. On CellProfiler 3, we had 1,700 features for all the channels, all the images, all the the objects. Sorry. And for the newest version, we have 4,200. So that's a lot more. And by checking the different versions, we saw that the texture is now measured in 4 directions instead of 1, and that the AreaShape also has some additional features being measured. So that's why we have this difference. And for the Endeavor, we have a bit less, so 3,300, because we are missing the RNA channel here. And then what we can do, we can aggregate this data. So if you take compound A, for example, we are going to take the single cell measurements, aggregate into replicates, and then finally aggregate into a medium profile, which we can then have the values for each feature with their respective medium values. So these features, of course, we have a lot of features, and we know that most of them can be redundant and do not add anything to our analysis. So we are doing feature selection, of course. And I chose this method that we were using in our the current workflow that we run to perform this analysis. But of course, you can choose different methods. So in this case, we are taking the replicates for a compound. So the replicate 1 and replicate 2, so 2 different plates. And then you take the median cell area, for example, plot all the values and measure the correlation. So if the correlation is higher, this means that this feature is highly reproducible between the replicates. Yeah, so these are called the robust features that we selected only once and resulted in 579 robust features. And this was done a few years ago. Axel performed that, and these robust features are based on our current imaging system, of course, and also in the current version of CellProfiler, the oldest version of CellProfiler, which is the 3.0. And then finally, we can have the consensus profile. We can also calculate the z-scores. This is how we usually compare the different samples. So you take each feature and then you calculate the median of the DMSO controls and then the median absolute deviation to these controls. And then, of course, you can have here, for example, nocodazole, and you can compare all different compounds that we tested with a reference compound and then check what are their similarity, what is the activation values for these compounds. And this is quite helpful. This Axel also developed a few apps that we can dynamically explore the data, which is quite nice. And then the induction values, they are calculated based on the number of features with absolute value higher than 3. And this is divided by the total number of features. And we have a threshold of 5% to say if the compound is active or not. And then the biosimilarity, which is basically a Pearson correlation between the compounds. And we do have a threshold of 75%, or you can go up a little bit more if you need to be more precise. So if you have, for example, compound A, compound B, and you plot all the feature values, and then you check what is the direction of those features, they can be highly biosimilar or less biosimilar. Yeah. So this is our database. These are the reference compounds. This is just an illustration of how many compounds we have tested, at least the reference ones. And these are the biological clusters that we have. Not all of them are published yet and validated, but they're on the process, of course. And we have already measured 5,400 compounds, which are reference ones. Of course, there are more. But then from these ones, 30% of them are active in 2 and 10 micromolar concentration. And from those, 80% have biosimilarity higher than 75. So we can classify at least 30% of the compounds. And for this test with the Endeavor, we have a pretty small, I would say, data set, which we are working on expanding now that we have the system on site. So for this test, I had to do some filtering. So I'm using only compounds that have more than 75% biosimilarity, and compounds that are reference, of course, which has known mechanism of action profile. And then here you can see our published clusters. So here's a UMAP feature reduction. And then you can see all the different biological clusters that we have. And yeah, I would like just to, so you pay attention to the DNA synthesis and the aurora. So those are both, there are compounds that can act on the cell cycle. And this belongs to both of these clusters. And we want really to separate them because sometimes they can cause phenotypic alterations on the cells, which are can seem similar, for example, in large cell or multinucleated cells. And what we want on the Endeavor analysis too is to have, for example, those two clusters well separated. So yeah, also how we annotated the compounds from the images with the compounds from Endeavor is taking the ground truth labels, which were defined with our previous imaging system. Yeah, finally, how did I analyze this data? I did some UMAPs, and then, of course, I used k-means clustering, which I think is the most straightforward one. You can also choose other methods, of course, of unsupervised clustering to compare the ground truth labels with the predicted ones. And then I ran the experiment at least 100 times so we can get a mean adjusted rand index value, which is equal 1 for a perfect match or minus 0.5 for a discordant cluster. Yeah, now the first question is, does CellProfiler affects version affects image analysis? And to do this analysis, we found the 579 features, robust features, which are equivalent in the newest version of CellProfiler. And then I use the same images as a source, and we hope that they should be highly correlated. Of course, if you plot the cell area from the same images, same source, you would expect them to correlate, I would say at least 0.9 above, because this should stay the same, right? And this actually happens, okay? But for some correlation features, they do have low Pearson scores. So on CellProfiler, you can add different modules to your analysis. And this module called Measure Colocalization does have a few discordant features. So for example, even negative related ones. And this can be connected to some changes on how they process the images on the new version. So this is explainable. But yeah, this is just something that I wanted to bring up if anyone is using CellProfiler and wants to know. And yeah, now for the next two questions that I'm going to answer together. So clustering with 4 channels is enough, and should the feature set stay the same? Yeah, so for Endeavor here, you can see the different clusters, and actually it does pretty well. You can see, for example, the Aurora and the DNA synthesis cluster, they are well separated. The other ones, they're a bit closer, but this we also see with the CellProfiler 3. And if you compare with our prior imaging system, yeah, we have, I would say, similar results. These ARI values are pretty close. And we also have a nice separation between the different clusters. But this imaging system was including the RNA channel, right? So what if we remove this channel and check the performance? It actually, I would say, stayed the same. It's pretty close to, which indicates to us that this RNA channel is actually very redundant. And I will show you why, because if you do an overlay of the endoplasmic reticulum and the RNA channel on our prior imaging system, you're going to see the overlap. So here in orange, yellow, inside the nuclei. So clearly this cyto14 stain is leaking also to other channels. And the same thing happens with the Endeavor. We are not acquiring exactly the cyto14 on Endeavor, but when we acquire the ER and the actin, Golgi, and plasma membrane, we still get the same pattern. This is nucleoli inside the nuclei. So I would say going forward, we would keep doing the same panel of stainings because this just makes sense. And you can still have all the information here. Yeah, on using Endeavor with 4 channels, you have less channels, so this is good. Yeah, just an observation. And yeah, and what if we add more features? I think you notice that we went from 3,300 features to 251. And it's quite a low number, but they all are reproducible between the repeats. And if we add more features, actually also doesn't make a lot of difference. Just a little bit on those 2 clusters, they are now more closer together, but still I would say it's good. So yeah, basically cell painting is very robust. You can do a lot. But you need to have the right set of features, of course. Because if someone was asking, if you try to use the 579 features that we use on CellProfiler 3 to aggregate the data on Endeavor, we actually got a pretty random cluster. As you can see here, nothing's clustering. We have an ID of 0.06. And so, yeah, it's very important when you change imaging systems to check if the same set of features works or not, if you need to do, again, the feature selection. So this is going forward now. This is what we want to actually got into a consensus of how we are going to analyze the data and which features we are going to use with the Endeavor. And now, so if you take a look on the feature types. So yeah, CellProfiler can export, for example, area shape, texture, intensity, radial distribution, granularity, correlation, and neighbors. These are the main, I would say, measurements that CellProfiler can do, the different modules. So if you take a look, the Endeavor selection of features, we have most of them belonging to the area shape, so related to the morphology of the cells. And in second place, the texture. And in our current pipeline, we have the texture as the main source of the features, and then the other ones. But yeah, then you can get into a whole discussion about if also you change the system, so now you have different cameras, different objectives. So this, of course, will impact on your feature selection. And yeah, another thing that we tried was to do this simulated binning. I just want to reiterate, oh God, that this binning method actually is an underestimation of what the microscope can do. Because if you take like the real binning on the hardware of the microscope, this is better than the simulated one. So this is even an underestimation will result in good results, right? So we can see that still we have this nice separation between the clusters, and that we can, easily acquire these images on 2x2 binning, which reduces a lot the storage that we need to have. So this is very good. So we do have more, of course, data that we are acquiring now, and also with 2x2 binning. And so stay tuned for updates. Yeah. So what we learned, we compared the prior imaging system with the new. We know that moving to CellProfiler 4 is going to happen because now, of course, we want to update our pipeline, and also we need that for the new images from Endeavor. We know that 4 channels are enough, so we want to keep, I would say, acquiring cyto14 because we still have the information about the RNA, which is good. And the feature set is yet to be determined. When we acquire more data, then we can actually get into a consensus about that. And then, of course, 30 times faster, the Endeavor. So really helps us on the day-to-day tasks that we have in the lab. So this is quite nice. The area coverage also helps a lot. So we do acquire more cells, which is, important for a more robust morphological profile. Also, we are acquiring fewer channels, and it does not affect the CellProfiler assay performance. And finally, the expand usage. So we are actually also using the Endeavor for other assays. So, for example, glioblastoma assays that I'm doing, some live cell imaging, and it quite helps on the day-to-day basis because you just go there, take a preview of your plate, and actually I'm using basically every day now, so it's quite good. And yeah, I want to acknowledge, of course, all my colleagues. Without them, we could not do this work. Also, of course, acknowledge Axel for the apps and the cell painting workflow, and also our funding agencies, of course, and the COMAS and the Max Planck Institute. Yep.
I have 5 more minutes in case you wonder what this Endeavor is she's mentioning and what it looks like. And we actually had a big announcement yesterday. We launched 2 more products here at SLAS. The Endeavor Pro is what we have at the COMAS. And we now launched Endeavor Ultra, which can, in some cases, go twice as fast. And we have a more budget-friendly Endeavor Core that is a bit slower, has less of the imaging cores inside, but gets to the same image quality as the same easy-to-use software. And with these, we have, of course, the Endeavor acquisition software, which is, I hope you would agree, easy to use. We have the clairvoyance analysis software, which was not used for this, but can be used for a lot of other assays. And what we introduced with Endeavor Ultra now is the Clear RT for real time, which gives you a quality readout while you are acquiring the images. So while the plate is still in the machine, you get a readout of your cell count, your signal to background, all these kinds of things, focus quality that you want to validate while the plate is still in there, maybe take action on these quality readings. So use case for the Ultra, of course, 4 minutes per plate. I used to say the Pro is the fastest high content imager on the planet. Now the Ultra is. Use case, of course, you have a huge library. You have all these CRISPR things to edit genes. You have all these compounds to screen, or you're making your own compounds. It's the fastest thing we can offer. Of course, it's automation ready with a CELA2 interface like the other 2 units. But this would be the fastest offering with the best QC that we can bring to you. That's the new system. Then the Endeavor Pro is actually a system that was used for all the cell paint. And it's a system I have live at booth 612. 612. Says Endeavor multiple times because we have 3 of them there. And one of them is a live unit. And I can show you the speed and the image quality if you want to come and go there. And of course, application here, live cell assays, because the plate goes in, you do a fast scan, plate goes out, back into the incubator. Cell painting for medium-sized libraries, I would say, these kind of things is what you would use the Pro for. And then the core with the reduced imaging units, but still giving you the same quality, more budget-friendly. Maybe if you have a lot of small runs that you have to do, or if you're just developing the assay and don't want it on automation, you want a walk-up system for that. Or if you're doing QC after you have found your leads and you need to validate and then come back and only have, 2 plates a week, then you don't need the Ultra, but we have the core for that. Gives you the same images. You can't tell what system they're from. It's the same sensor. It's just the underlying infrastructure that makes them a lot faster. So that's it from my side already. And thank you for being here.